Okay, welcome back to the second half of our event tonight. On behalf of the PTFI, I am Tracy Shafazada. I am with the Verso Biosciences team, and I am the Director of Programs and Partnerships. We're going to kick off the next panel now, talking about the technology underpinning the PTFI and the development of the database. We're going to move on now to share remarks from the Honorable Daniel Glickman, who is not with us today, but we are going to show a quick video that he has prepared for us. The Honorable Dan Glickman is a past chair of the Board of Directors at the Foundation for Food and Agricultural Research. Mr. Glickman served as a U.S. Secretary of Agriculture from March 1995 until January 2001. Before his appointment, Mr. Glickman served for 18 years in the U.S. House of Representatives, representing the 4th Congressional District of Kansas. Currently, he is the Vice President of the Aspen Institute and Executive Director of the Aspen Institute Congressional Program, which is a nonpartisan public policy education and civility building program for members of Congress.
Hi, I'm Dan Glickman. I am the former U.S. Secretary of Agriculture and the founding chairman and current board member of the Foundation for Food and Agriculture Research, one of the partners in this effort, the Periodic Table of Food Initiative. So I'm so delighted to be here as part of this effort and to support the effort, and delighted that the foundation, or as we call it, FAR, is participating in this great project to pioneer the future of food and agriculture and human health research. And what this effort will do is the research will guide governments, the private sector, and non-governmental organizations, and the health and medical community as well in developing better food policies that reduce hunger and malnutrition. You know, one of the things that I have thought about over the years is that 3 crops, rice, wheat, and maize, or corn, as we would call it, account for 2/3 of the world's food supply. But we rely on those very few crops, and we should broaden that rather dramatically to improve the world's nutritional security, which, and this initiative will reduce the threat in various ways, largely by looking at all crops, particularly underutilized and local crops, looking at a crop's functional ingredients, and looking at the whole issue of diversity and biodiversity of crops as it relates to nutrition, all to perhaps power a new world where what you eat can determine your health with much more impact than it currently does. So this initiative will help to do this. Hopefully, it will unlock the unknowns of ingredients of food, which will impact human health by providing the data that we need to create preventative and proactive and protective diets that are based on robust scientific evidence. The results from this project will help nutritionists and medical scientists design and customize diets to meet nutritional and medical needs of individuals, or design healthy food programs using region-specific food. The knowledge will help farmers and ranchers and livestock producers and producers of all commodities all over the world get the kind of knowledge that they need to improve their crop production techniques and to produce healthier foods in the process. We hopefully will help us create new national and international health programs and influence government dietary guidelines, because we will know more about the composition of foods and not just accept what we have been told to accept over the last decades or centuries, but really try to analyze what's in our foods in the same way that we do analyze what's in our drugs and pharmaceutical items. And this initiative will finally determine the benefits of underutilized or neglected regional crops that can help nutritional security in largely the developing world where they need it so dramatically. So this initiative, it's so important. It will give us the data to understand food. I think we understand it because how it tastes, but we need to know what its impact is on our bodies and on our environment. And this initiative will help move us very dramatically in that direction. Thank you all very much.
Great. Okay, I'm going to invite Dr. German up one more time to give some brief remarks on his vision for the PTFI and the technology that we're building around the PTFI.
All right, now I want to talk to you about the impact of this database that we're proposing on all aspects of the scientific process and its utility. And I want to give you a couple of examples of why we're so confident that this is going to disrupt and improve the human condition. So one of the things that building this database will do will allow us to understand, to discover how food works. And we are confident of that because that's what we at UC Davis have been doing for years with human breast milk. So by taking this comprehensive approach to what's in breast milk and what does it do, it allows us to understand lactation as a model. And lactation is a model of all aspects of what we're pursuing. It's, in essence, our model is the mother-infant pair. The mother is literally dissolving herself to make a comprehensive and complete diet for her infant. Everything costs the mother. It's a model of sustainability, like you have never thought of before. And yet, everything in the milk is nourishing that infant. If it doesn't help the infant, it's going to be lost. Evolution will not reward mothers who are producing something that's not valuable for the infant. But if anything in that milk is valuable for that infant, it's hard to imagine anything under more positive selective pressure. So that's what we've been trying to figure out, looking at every component of milk. And it's so much more than just the essential vitamins and minerals. Every baby needs every single vitamin, every single mineral. Of course, milk has that. But milk is so much more instructive. So let's take protein. We're very concerned as a world, will we be able to make enough protein for the world's food supply? Let's ask your mother. So in fact, is human milk teeming with protein? In fact, babies are building themselves with protein. In fact, milk is not extremely high in protein. What it is, extremely rich in the best of protein. So if you look in detail at protein, the composition, the sequence, they are all ideally constituted for the infant. So all the amino acids are necessary for the growth, all of the, in essence, components. But mothers do one more very clever thing. Digestion of protein is actually not easy to do. How does milk solve that? Mothers put enzymes in the milk, and the proteins, in essence, are activated to be digested in the baby. The mother is feeding forward. Can you imagine that the future of food will include the proteins and the ways to digest them? They're optimally, in essence, harvested and processed so they're optimally digestible. That, in fact, will change the way we think of the amount of protein we need worldwide. Second, milk has fiber. What? Why would mothers dissolve themselves to make undigestible matter in their milk? Why would they digest themselves to make poop in their baby? Made no sense. But we took this fiber material apart. It's small chunks of fiber that the mother actually synthesizes. The baby can't digest them, so they go right through. What do? In fact, they don't feed the baby at all. They feed a very specific bacterium in the baby, and then that bacterium populates the lower intestine, the microbiome of that infant, protects the baby from opportunistic pathogens, and nourishes and fuels a part of the baby that milk has difficulty getting to. What an ingenious idea. So, could our foods of the future feed us and feed our bacteria in a very specific way? Of course they can, and of course they will. Second area of science that will explode with this is population-based studies. So now we already know that different populations suffer different health outcomes, and we've recognized that's associated with what is called patterns of consumption. What that really means is no one food is important over another. It's the diet that we have to consider. But if we only know them as food patterns, we can never translate that. Once we know the pattern in a molecule-by-molecule ensemble basis, then we can take the knowledge of any population in the world and then transfer that to everyone. So everyone can take advantage of pattern recognition for our diets. A spectacular future. already imaginable. But the real, in essence, change we will make agriculture, food, diet, a knowledge-based system. And that sounds a little bit esoteric. What's a knowledge-based system? Well, actually, you all know what a knowledge-based system is. Transportation, Google Maps. So in order for Google Maps to work, there had to be a massive database of all the roads and highways, bike paths in the publicly accessible cloud. That database is there. And now, your individual personal device, equipped with simple algorithms, can interrogate that database to answer a very simple value proposition. Where am I now? Where would I like to be? How do I get there? And with that simple value proposition in place, think of what's changed. Of course, we don't sit in a car and open up a map when we're lost. We're guided to our destination. But then it was recognized that much of the world depends on knowledge of the transportation system for transporting people. Taxis, obsolete Uber. Delivery, obsolete Amazon. Hostelry, how do you find a destination? Well, device, Airbnb. We have changed the world by a simple knowledge-based system. And then, obviously, what's next? Autonomous vehicles. So the knowledge base associated with just knowing where we are is changing the world around us. Now, take food, the database of food. What's the value proposition? What's my health now? What would I like it to be? How do I get there? That's a magnificent future, and that's what this database will make possible. Thank you.
I hate to do that. I absolutely love listening to Bruce inspire all of us on food and nutrition and health. I'm sorry, Bruce. I'm now going to introduce Dr. Steve Watkins. He's the final speaker on this panel. Dr. Watkins received his PhD in food science from the University of California, Davis. He is currently the founder and director of Verso Biosciences and the CEO of BCD Bioscience. Steve?
Thank you, Tracy. All right, well, as a designated technologist, I'm the only one to have brought slides, so I think that's, anyway, I am very excited and actually probably more grateful than anything else to be a part of this initiative. I think it's coming at a really important time, and it really seems committed to tackling the big problems with analytics that have hindered our ability to generate truly useful data for nutrition, and I'm very happy to be a part of it. So, we good? There we go. Roy mentioned earlier that, surprisingly, a large portion of the molecules in food are unknown to us. Right? There's a lot of dark matter out there, and we don't even know what it is. So this initiative is absolutely committed to shedding light on that and to beginning to identify new food compounds. But I thought I'd point out that even for the compounds we know exist in foods, things like macronutrients and their derivatives, the derivatives of amino acids, derivatives of fatty acids, and et cetera. For secondary metabolites from plants, flavonoids, terpenes, or synthetic compounds or natural compounds that are involved in herbicides or exogenous chemicals that we add to our agricultural and food supply. We know they exist, we know they're in foods, but we have no tools to quantify them, to catalog them, to actually build reference resources for them. So there's this parallel needs. One, what's out there in the universe? What do we need to learn about in terms of the composition of our food? And how do we begin to develop technology that can catalog these compounds so that we can understand them and do research and understand how to manage our futures? So the mandate for the technology part of the PDFI is very clear and very exciting. It really has a couple of parts. One is to build the world's best reference database of food molecular compositions. Right? So we're building a reference database that will allow everyone to understand a lot more about what is in their food and how it changes with different interventions, with different locales, different food types, et cetera. And simultaneously, to develop the technology that can begin to elucidate that dark matter, really discover and annotate new molecules in food. But the way we're going about it is what I think is really exciting, which is that we're committed to building the analytics, the technology that allows anyone to participate in the generation of this knowledge. So we're developing standardized, democratized platforms that can be distributed to labs or around the world to participate in the creation of this database or the creation of data that can be compared to this database. There we go. OK, so the technology really has three primary components. One is analytical chemistry. What are the methods and protocols and tools that we use to generate the data on food compositions? And so that's one big component of technology. Another is how do we begin to store this data, categorize it, present it to users of different types? There are a lot of different users of food composition data, everyone from discovery researchers in academia to governments trying to set nutrition guidelines for their populations. So food composition data and platforms and how you slice and dice that data and present it are an important part of it. And then there are strategies for actually mining the data that are coming in to really start to elucidate this dark matter. So we think about developing technology on all three of these fronts. And as Roy kind of stole my thunder, as is per usual, I wish actually I brought, like, we did strawberries, actually, we did spinach, we did a bunch of other foods. I should have brought you another example so you didn't think we just did apples. But what we did a study where we took blinded duplicates of the same food, and we sent them to a lot of different labs to get a landscape assessment of where is the technology today and what do we have to do to make it ready for the PTFI. And as Roy pointed out, what was really clear was that the platforms and technologies for generating data are very precise and reproducible within a lab. When we got data back from a lab on the blinded duplicates, they lined up perfectly. They told us they were the same compounds, same concentrations. It was all really nice data. But when we began to compare data between labs is when we saw that we had a real problem with harmonizing data into an integrated database. In apples, for instance, the labs returned data on 927 named compounds in foods, but they only agreed on the names of 14 of them. Okay? So some of those may be that they were actually measuring different compounds because they had different approaches. Others are that they were naming the same compound something different with their annotation strategies. So there was a lot of work to be done on harmonization and standardization, and that's really where we focused. So we're developing assays for small molecules, metabolites. These are things like plant secondary metabolites and intermediary metabolites like amino acids, et cetera. For lipids like triglycerides and phospholipids, fatty acids. Proteins and amino acids, vitamins and minerals. And as I mentioned, the platforms for measuring all of these components are actually fairly robust and do a good job. So what we did was we adopted existing protocols for separating molecules and measuring them. But we spent a lot of time designing and manufacturing chemical mixes, reagent mixes, that we'll ask every lab to put in every sample so that we can basically align the data sets that are coming out of labs from around the world. And as part of that, we're building centralized data processing tools that allow us to compare that aligned data and to do very consistent and robust annotations of it. So we can have a distributed analysis strategy, but a centralized standardization and data processing strategy, which allows us to do centralized data quality control as well. We can choose what has been done with a sufficient quality to bring it to the database. We can work with our satellite network of labs to continue to improve their assays. So this is actually, we have just rolled into kind of the beta part of this project where we're using these technologies to look at the first 100 foods that we've chosen. And we've chosen these 100 foods to represent a really broad array of types of foods for the platforms. And so the data are literally rolling in this week on a number of platforms. But the lipid analysis is finished. And the way you can look at this chart on the lower left is this is 1,300 individual lipid molecules that were measured in these 100 samples. And the 100 samples are aligned on the bottom. And then anything that has a purple color to it is really low abundance in the food, and anything with a yellow color is really high abundance in the food. And you can see I just highlighted a section of this plot where you can see various foods pulled out with quite distinct lipid compositions coming back from the different foods. Brussels sprouts, for instance, had a really high proportion of lipid species that contain long-chain omega-9 fatty acids, which is unusual. It's common for brassica, but they have been bred out of a lot of brassica, but Brussels sprouts still contain them. Beef liver has lots of inositol lipids and serine lipids that are unique, and you don't find them in many foods. Cheeses, beef, coconut, obviously lots of short-chain fatty acids. I thought it was interesting that red and brown lentils, which we think, I don't know, I'm a lipid person, I wouldn't think they had a really distinct lipid composition, but they clearly did. They had a number of lipid components that were quite distinct from the rest of the food. So really fine detail, a lot of information that we'll be unpacking for years to come, just on the basis of running these 100 and next couple thousand foods. All right, so the strategy and sort of where we are in the timeline, we worked really closely with a small set of labs that have great protocols that we've adopted and modified with our reagents and central data pipeline. That was phase one. That was really in the establishment of the methods and protocols that we'll use in the larger project. Right now we're at phase two, where we're beginning to transfer those protocols to a set of labs around the world, some of whom you'll meet in a couple of minutes, to run food samples in a distributed but controlled set of labs around the world. And we'll continue processing these inspirational foods over the next couple of years. And what we envision, though, is the next step beyond that is to make these reagents and the data pipelines available to any lab around the world that wants to participate in this project so that we fully democratized the generation of these data. And I'd just like to quickly point out that it isn't lost on us that ultimately we need to bridge the gap between food composition and what it actually means for human health. And the great thing about these platforms that we're developing is that they're equally powerful at actually generating data on human samples, human plasma, serum, fecal material, urine. And so part of our work is also enabling the use of these platforms for human samples so that we can begin to do some translational work, really understanding how food composition relates to changing composition in human samples. And with that, thank you very much.